This is the growth rate here, and here is the temperature, okay? So certain bacteria would have their optimal growth rate at temperatures of around 100 degrees. And those are called hyperthermophiles. So they like the heat, and they have their optimal growth in this heat. Some will have their optimal growth around 60. A lot of the bacteria that we have in our bodies have their optimal growth rate in a range between 30 and 40, around 37, like those mesoforms. So temperature is important in the growth of bacteria, and you can control for that in the lab by having incubators. This is what happens in microbiology labs. You have incubators that control the temperature. They control the CO2, which plays a part in the control of pH of the culture, and they also control the O2 concentration. So you set all those parameters in the incubator, and you adjust it according to the bacteria you're trying to grow. Now, if we are talking about media, we should know about selective and differential media. So selective media, as the name implies, selects for certain bacteria to grow in them. So it does not allow the growth of any bacterial species that you put in this media, only certain bacteria. And this selectivity can be done through having certain nutrients that only this bacteria can use, or by having, for example, an antibiotic that would kill all other bacteria except the bacteria that you are looking for. Okay, so this is selective media. While in differential media can grow several types of organisms. It's not selective for a certain type of organism. But how the organism reacts to this media can tell us what organism it is. And let's look at this example here. This media is called the blood agar. So we made this media called blood agar. We pour it in 3 different plates, and then we grow our culture. And depending on the pattern that we see, for example, of hemolysis here, we can tell what bacteria it is, okay? So because this medium is made of blood cells, we can see that different bacteria can lyse those blood cells in different ways. So for example, if we see this pattern called beta hemolysis, we will think that, okay, this is Streptococcus pyogenes, because we see this pattern. While if we see this pattern, we will think that it's a different bacteria called Escherichia coli. So the differential, this differential media helps us know what organism we're dealing with, depending on how this organism interacts with the media it's put in. Okay, does that understand a bit? The difference between a selective and a differential media. Okay. So here we come to the last slide where we discuss how we can isolate bacteria into pure cultures. So in order to study the properties of a certain bacteria, we need to have it in a pure culture all by itself, so that it's not with other bacteria that can influence the characteristics that we are going to measure. So for example, if a specimen comes to the hospital in the microbiology lab, for example, a urine specimen, it will contain maybe several types of bacteria. But in order to know what types of bacteria, and then later on choose the bacteria we want to study, we need to have single colonies of that bacteria. We need to have a pure culture. And that pure culture will come from single colonies. Okay? So here, someone has dipped this inoculum. See this inoculation loop, sorry. See, this is called an inoculation loop. They have dipped it in the specimen first, and then they came on the plate and made streaks. Okay? Then they put the inoculation loop on fire so that there's no more bacteria on the inoculation loop. And then they take from this streak another streak. And so on, they take another streak, and then from this streak they make another streak. So it's simply a process of dilution. You're just diluting without actually making numbers. You're diluting the original amount by just making streaks in hopes of getting an enough dilute streak here, finally, that it will give only single colonies. You see, this streak has a lot of bacteria. It came directly from the specimen. It has plenty of bacteria. But this streak has less, and this one has less. And finally, this streak provided us with what we were looking for, which is single colonies. And those single colonies then, we will take with the same, with the inoculation group, not the same inoculation group, sorry. The next day, when we come, we'll see single colonies, and we pick those single colonies and grow them in what we hope is a pure culture. If we did not find single colonies, we need to repeat the process again until we get a pure culture of homogeneous colonies. Then we can study, for example, the antibiotic resistance of that pure culture. Yeah? This can also be done by another method other than streaking. We can do the dilution in another form. For example, we do the serial dilution, which we have talked about in the start of the lecture. We do serial dilution so that we don't have a lot of colonies stuck to each other. Yeah? So we can separate the colonies. We do serial dilutions, and then we add the diluted specimen to agar just before the agar solidifies. So we add it to liquid agar, and then we pour it in a plate. And then the bacteria will be trapped within this agar, and then hopefully if the specimen we had was dilute enough, we will be able to distinguish single colonies from different bacteria. So this is a result, for example, from this streaking. This person has streaked the specimen he had and ended up with this plate, and if you zoom in, you can see that he did end up with single colonies from different bacteria. Yeah? Because colony morphology, how the colony looks like, is reflective of what the bacteria that formed this. So, for example, this colony is formed by a different bacteria than this one. And this one is probably formed by a different bacteria than this one. Okay? So, the microbiologist will look at this and then choose a colony that he suspects is the pathogenic, for example, bacteria within this culture, and then try to grow it in a pure culture so that he can learn, for example, the antibiotic susceptibility of this organism. And as you can see, if you get the sample directly with mixed organisms, you cannot test it for antibiotic sensitivity. Yeah? And you cannot test other parameters because it's a mix of bacteria. So we need those pure cultures in order to study the bacteria we want. Yes, this concludes our lectures, so I will need some questions now. Yes? Yes, you can do that. If you can do them in combination, you know. It's a group of methods that you're going to use in order to get the bacteria you want. So, for example, you can use the selective media, and then you could end up with the bacteria if you were correct, the bacteria you're looking for, or you can end up with no bacteria growing on the culture. So what you can do is try and see what bacteria you will have, and from the colonies, by the streak method, and from the colonies you can then have a more educated guess on what you can do next, what selective media to use, what differential media, and so on. Yeah, you need a single colony in order to get a pure culture. Yeah, you can like if you have 10 colonies of the same shape, you can then study it as a pure culture. Yeah. So selective media only grows specific organisms that you are selecting for, either through nutrients or through antibiotics, for example. So you have a certain selective criteria that gives you an organism. While in differential media can grow more than one organism. It's not selective for any organism. But through knowing the interaction between the medium and how the organism interacts with the medium, we can tell what organism it is. Okay? So differential media depends on how the organism interacts with that medium. Okay? Is that clear? Yes, any more questions? I understand this might have been a difficult lecture. No? Any more questions? Okay. Finally, this is the further reading you can do from drawings. And also, there's something I found called agar art. So you can actually draw an agar by knowing what bacteria gives what colonies in what shape. You can use it, for example, a golden bacteria to form this part of the drawing, and then another bacteria colonies that give different colonies, and so on. And you can see all of those are agar drawings. And the American Society of Microbiology even have a contest for this. So if you have some artistic sense, we can provide you with an agar plate, and then you gather some knowledge on how your art can look like. You can enter that contest. Yes? Any questions? Better to ask now than after the lecture. No? Okay, thank you.